Good evening, everyone. So, in today's class, we'll look at a sample questions to help you think about week three's lectures and solve the assignment. The first question is the most appropriate technique to study DNA protein interaction is. Four different options are given here: hydrophobic interaction chromatography, gel mobility shift assay, STS page, and option D is Western blot. Whoever knows the answer can think about the question and you can start raising your hand. You can take a couple of minutes to think about the question. The important thing is do not tell the right answer. Instead, we discuss all the wrong options first, eliminate why they are not appropriate, and then come to the right answer. You can think about this question for a couple of minutes and then raise your hands, and then we start discussing. Even if you don't know the right answer, if you think one of the option is incorrect, you can still try mentioning which of these is incorrect. We can take it from there. So Preeti and Shanmugapriya have mentioned options. You can unmute and justify why you think that particular option is correct or incorrect. First, you can explain what that option is and where that particular method is used and why you think is right. Yeah, one of you can unmute. Maybe Preeti first.
Hello, sir.
Yeah.
As as we have learned previously, also southern hybridization for DNA interactions, northern for RNA, and western for protein interactions. That is my answer is western blotting.
Okay, so can you briefly summarize what happens in Western blotting?
we just take the that gel and just analyze in this Western blottings along with the DNA probes.
Okay, so Western blotting is a method where we want to analyze specific stretches of amino acids, like small peptide region or a protein itself, for which we use an antibody. And this antibody is linked to another readout, similar to how people do for ELISA, but this is done with uh, a, a page, usually an STS page, because you have better resolution there. So there you can't really study DNA protein interaction. Uh, yeah, it is not a possibility for you to study DNA protein interaction there. There you can just confirm the presence of a particular protein. Like for example, people generally use Western blotting as a method to detect if uh, a protein with a particular tag is present. For example, proteins can be expressed with his tag, with strep tag or S tag, any of these tags that are naturally present in pet vectors. So antibodies raised against these small regions can be allowed to interact with the proteins on the blot, and we can visualize them later. So Western blotting is used for that. It is not a technique that is meant to study DNA protein interaction. So B is incorrect.
Okay, sir.
Shanmugapriya, can you explain what hydrophobic interaction chromatography is?
Sir, it was just a guess, sir. Exactly.
Okay, okay. So this is one another method where we can purify proteins based on how hydrophobic these are. So proteins hydrophobicity is just determined by the amino acids that are present in it. It is not very specific. Similar to your anion exchange and cation exchange, this hydrophobic interaction chromatography also separates proteins based on how hydrophobic that is. Both these options are incorrect, and the third incorrect option is again STS page, because of the nature of the page, which is it is denaturing. You will not be able to confirm whether uh, DNA protein interaction is actually happening. What you will be able to see is uh, a protein in the absence of DNA binding will run at its actual molecular weight. Protein in the uh, presence of DNA will run at a higher molecular weight, because it is now bound to DNA and it cannot move faster, so it will be retarded, its motion will be retarded, so you will see it at different levels. But this cannot be done in STS page, because in STS page your samples are denatured. Only in native page this can be done, and the method that I just explained is called gel mobility shift assay. So here if you see, uh, we have DNA that runs till this uh, point, or this length of the gel. Then you have DNA plus protein that doesn't run till the extreme end. Here you will see some DNA depending on what stain you use and what gel you use. If your stain is ETBR, that is expected to bind to your DNA, then you will see only DNA here and DNA here. Whereas if you use a stain that binds to your protein, for example, Kumasi brilliant blue, like we discussed earlier, you will see a protein band here and a protein band corresponding to the free protein. What we see here as the third part is the antibody. So this antibody is nothing but an antibody specifically raised against this protein. And now, because this is of even more higher molecular weight, we see an increase in the shift. So this distance between your DNA and the DNA protein complex is called shift. And because you observe a shift in the bands, they call it gel shift assay, mobility assay. So the answer is option B, gel mobility shift assay. The next question is the atom radioactively labeled in DNA to enable us to visualize it in autoradiogram is option A phosphate, option B lithium, option C sodium, option D magnesium. You can think about this question for a couple of minutes and then uh, um, yeah, raise your hands. Yeah, Preeti, you can unmute. You can unmute, Preeti.
Yes, sir.
Yeah, you can mention what you know about this question. You can briefly describe why you think phosphate is the right answer.
Yes, sir. We have learned that we are using phosphate, like the isotope P32, because it kind of intercalate in the DNA backbone.
Does it intercalate or
Like incorporates into the backbone.
right? So it becomes a part of the DNA, right?
Yes, sir.
Right. Yeah. So there are few elements that are a part of your uh, DNA, which includes carbon, hydrogen, oxygen, nitrogen, and phosphate. Phosphate is the one that links two different bases. It's called a phosphodiester bond, and when you have a isotope of phosphate which is radioactive, it is easier for visualization. This is how earlier it was done. There is no concept of intercalation here. Intercalation happens when you have something like ETBR, yeah, yttrium bromide, which goes and stacks between two nuclear bases, because of which it has enhanced fluorescence. But yeah, for this particular question, phosphate is the right answer. The next question is the blocking agent used to prevent non-specific binding of protein during footprinting assay is option A PMSF, option B DDT, C Salmon sperm DNA, option D BSA. So for this particular question, I would want uh, specifically I would want all the wrong options to be eliminated. So before anyone goes to type out the right answer, you can unmute. I mean, first think for a couple of minutes about each of the option and first describe what a footprinting assay is. Then we'll go discussing about each of the incorrect options and finally to the right answer. You can take a couple of minutes to think about this question.
So the footprinting assay is like when binding takes place between the complementary sequences. Is that like in case of the southern hybridization, we use solvents from DNA.
No, no. So we can use salmon sperm DNA for multiple different cases. But what is a footprinting assay? Do you have any idea about what a footprinting assay is?
Might be I'm wrong. I don't know, but the nylon membrane we are using for that we do.
No, nylon membrane can adhere to specific molecules of specific charges. Uh, can you try thinking just from the name of it? Footprint. Does it make any sense to have a footprint and what it would mean in a molecular biology context?
As if like blocking of that membrane.
No,
That we are saying.
that is not footprinting. You can maybe think. We'll check with Meenu first and then Sai Sampath next. Yeah.
Yes, sure.
Yeah, Meenu, you can unmute. Yeah.
It is a technique used in molecular biology for identifying the specific DNA or RNA sequences where the protein binds.
Right. So you have a DNA or an RNA binding protein and you want to identify, pinpoint which particular sequence within the entire DNA or RNA where this is bound. That's what you're saying. Yeah, that's the right answer. Right. Good. Now, who was it? Sai Sampath. Yeah, you can unmute and tell what other information that you know about footprinting assay.
The same answer, sir. It is easy to find where the DNA RNA sequence binds the specific protein.
Right. So now, just adding on to whatever was said, why would we need a blocking agent here? It is there in the question itself.
For non-specific binding?
Yes. So you will have multiple, in many cases you will have more than one protein and more than one stretch of DNA. You don't want these interactions to be random. You want to identify the specific protein's interaction with a specific DNA. So you try to ensure that no charge based random interactions are captured during your effort to identify this region, which is why you try to block. And for that particular purpose, people use this salmon sperm DNA. Not just in this particular work, in any kind of work where one would want to ensure that DNA is not um, the DNA of our interest is not randomly getting bound to a particular region. People use salmon sperm DNA. One another example that comes to my mind when I talk about this is when transformation is done in yeast cells. So transformation is this process where you introduce DNA, and when in usually when in this process you add DNA to a solution containing cells. You don't want your DNA to be bound to the exteriors of the cell. You rather want it to enter the cell. So what you do, you add salmon sperm DNA which coats on the layers of the cell, thereby preventing your DNA of interest from sticking onto the surface. Then it would efficiently enter. So the right answer here is salmon sperm DNA. But before we discuss this right answer, does anyone know anything about the other three incorrect options here? PMSF, DTT, and BSA?
Isn't the BSA works?
Protein degradation. It is used for protein degradation.
Sorry, can you repeat?
PMSF is used for protein degradation.
Do you want to degrade protein or can you expand PMSF?
It is a serine protease inhibitor, sir.
Right, correct. So it's a serine protease inhibitor. So what it does is when you add this to a solution, all the serine proteases, which is proteases that contain serine at its active site, they will be covalently bound to PMSF. PMSF, you have the full form in the next slide. It's phenylmethyl sulfonyl fluoride. So it binds to the serine in the catalytic triad. You might have heard about catalytic triad and enzymes. So these serine proteases have this catalytic triad at a particular orientation. PMSF covalently binds to the serine in the catalytic triad. So your proteases will not be able to cleave any of the proteins. So you will ensure that whatever protein that you have is not degraded. That's the role of PMSF. Good. What about the other two, DTT and BSA?
BSA also works specific protein, so it to the answer.
Sorry, can you repeat?
Then I am saying like BSA also works for protein protein binding blocking.
No. What is BSA? Can you expand BSA?
One serum albumin.
Right. It's an albumin from the serum of cows. That's why it's called bovine serum albumin. So this is a pretty non-specific protein. People generally use this in solutions where you want to store uh, your other enzymes. Like for example, any restriction enzyme that you contain will have a trace amount of BSA, because in case of contamination with proteases, your BSA will be the first target. That is one particular application that I think of that I can come up with for BSA. And what about DTT?
It is a thiothiazole.
Yeah, what does it do?
It is simply a reducing agent which breaks the disulfide bonds, and that helps in the proteins in I guess reduced form.
Okay, where do you have disulfide bonds in proteins? Can you think of a particular amino acid?
Antibodies as a disulfide bond.
Yeah. That is an example of a kind of protein. I'm asking about a particular amino acid which has this sulfur, which can actually form this disulfide bond.
Cysteine amino acid.
Yeah, right. So cysteines form these disulfide bonds. So there are two kinds of disulfide bonds. One is intramolecular, which means there is more than one cysteine within your same protein. So these two cysteines come close enough together to form a disulfide bond. And this can clearly be confirmed in your electron density when you solve the crystal structure of the protein. The other kind of disulfide bond is intermolecular. You have one cysteine from one molecule of the protein, and another molecule of the protein also has the cysteine. Now, because these molecules are close enough in the solution, they form this disulfide bond. Now, why does one want to prevent? Okay. Now that I've told about this, by using DTT, do you want to prevent intramolecular disulfide bond or intermolecular disulfide bond? Yeah. Can you guys hear me?
Yes, sir.
Yeah. So I was asking if DTT is added to remove intramolecular disulfide bond or intermolecular disulfide bond. You can try mentioning, because anyways we'll be discussing and you will learn. That's the whole point of this discussion session. You can tell whatever comes to your
Since
mind. Yeah. Sorry.
since we're talking about binding of protein to something else, so can we say intermolecular?
Yes. So the problem with intermolecular disulfide bond is, if one molecule of protein binds to another molecule of protein, and this happens within the solution, your protein will start to aggregate. You don't want aggregation of protein. If your protein aggregates, then it will lose its shape, it cannot function, which is why DTT is added. So these are the three different options that we discussed, the incorrect options. PMSF, it's a serine protease inhibitor. DTT, it's a reducing agent. It removes disulfide bonds. And BSA, it's a protein that people generally use when they want to ensure that in the event of a protease contamination, the protein of your interest is safeguarded. Right. The next question is, the cellular extract you will use to identify transcription factors for a given promoter is dash. Option A cytosolic, option B nuclear, option C mitochondrial, option D membrane protein. So take a couple of minutes to think about this question and then you can raise your hands. We'll discuss. This question is still vague. There is more than one correct option here. So uh, you just need to justify why a particular option that you mentioned could be correct. But before we go to the correct option, you can just start with the incorrect options. That will be easier. Yes, first we'll discuss the incorrect option. One of you can unmute and mention what you think is an incorrect option. Preeti, do you say that nuclear extract is an incorrect option? You can first tell us what you think is the incorrect option, then we'll go to the right answer.
The cytosol extract is incorrect option.
Uh, why would you say cytosolic is incorrect?
Generally, translation occurs in cytosolic and transcription occurs in nucleus. You don't find the transcription factors in cytosolic extract.
Can you repeat it once again?
Transcription occurs in nucleus, no, sir.
Okay. What about cells that do not have a nucleus?
So Could you repeat, sir?
What about cells that do not have a nucleus?
Uh, Both occur simultaneously, like translation and
Yeah, coupling is there, that is different, but where does it occur? Like, for example, in cases like bacteria, like in E. coli,
yeah,
you
in
don't.
prokaryotes.
Yeah, in prokaryotes, you don't have a well defined nucleus, right?
Yes, sir.
In that case, what would be the answer? We'll go from step by step. We'll increase the complexity. In prokaryotes, what do you think will be the, where do you think this transcription factors would be available?
Oh, it is for prokaryote or eukaryote, sir, for this question.
This question is not defined. That's why I told that we have more than one correct answer. So
Okay, then NB will be right answer if I'm not wrong.
then, which option do you think NE is it? Nuclear extract is it? No, no. Okay,
Okay.
okay. I'll simplify it for you. Let's consider a prokaryotic cell which is devoid of any nucleus. In that case, where do you think you will find the transcription factor?
And cytosol.
Yeah. What other extracts will you have other than the cytosolic extract in a prokaryote?
So what's a what kind of?
So, in your prokaryotic cell, we know that we don't have a nucleus. We leave it.
Yes, sir.
Other than your cytosolic extract, will you have any other extract?
No, nothing.
Why not? You have proteins that are expressed and that receive signal from outside world. Where are these proteins present? How can a cell interact with the outside world? Is there something that is helping the cell to interact with the outside world? Its environment?
Yes, sir. For cell signaling.
Yeah, yeah. So, where are these proteins embedded? Are these proteins on the membrane?
Membrane.
Yeah.
Membrane.
So at the basic minimum, we would say in a prokaryotic cell we have two kinds of extract. One is the membrane, the proteins that are bound to the membrane, and the proteins that are in the cytosolic fraction, which are usually soluble. And your transcription factors in these prokaryotes are available in your cytosolic because these do not have a membrane tag and these don't adhere to the membrane. So that is accepted. Now from prokaryotes to eukaryotes, normal eukaryotes, let's say mammalian cells. Where do you think you will be able to find the transcription factors?
In the nucleus.
In the nucleus, right. Uh, so option B is also possible if it's a eukaryote. What about mitochondrial extract? Why was mitochondrial extract mentioned here? Any idea on that?
Even it has its own DNA can.
Right. So mitochondria is an organelle and it has its own DNA. So possibly transcription can happen there. That's also true. Which is why I told more than one option is correct here, depending on what your organ system, what your system is. Right. Good. The next question is, addition of antibodies into the gel mobility shift assay would result in option A, super shift of DNA band, option B, no effect to DNA shifting, option C, retardation of DNA band, option D, breaking of DNA protein complex. So think about this question for a couple of minutes and then you can unmute. Think about this for a couple of minutes. Yeah, Preeti, you can unmute.
Yes, sir. In case the answer would be first one is super shift of DNA band because when there is addition of antibodies, the molecular weight will increase. So that is why it will shift to the lower mobility.
Right.
And
Now,
that's it.
this antibody, would it bind, where would this bind to?
On DNA,
If the antibody is binding to DNA, how will we confirm that the DNA is bound to another protein?
sorry, sir. Can you ask again?
Yeah, yeah. So, you mentioned that the antibody binds to the DNA. But the goal of what is the goal of gel mobility shift assay?
The we check on the gel the size we can check. So if the size is larger after addition of antibodies, so
No, no, leave
it
antibody.
will
Take antibody out of the picture here. Why would anyone want to do this gel mobility shift assay?
like whether the DNA is binded to the protein or not.
Yeah, so whether your protein is bound to your DNA or not, that is the goal. So if you want to confirm whether DNA is binding to protein, or actually it's the protein binding to the DNA, that is what people usually use this for, not the DNA binding to protein. So if you want to confirm if the protein is binding to DNA, would your antibody be raised against your protein or the DNA?
I'm not getting some.
Okay, so um, what I'm although what you mentioned is right, here your protein,
Yes.
your antibody should bind to your protein of interest. You want to see a difference in how the DNA moves in the absence and in the presence of the protein. In the presence of protein, your DNA is shifted. That is what you see here. And in the presence of this antibody, it is even more heavier. So it is super shifted, like how you mentioned. But what you need to understand here is you want to track whether your protein binds to your DNA or not. You don't care about what happens to your DNA. So your antibody should be raised against the protein. It is something that you can remember. Was it clear or shall I explain that again?
Sir, once again.
Yeah, yeah, sure. So, like, I think you guys understood that when you add antibody, it is heavier, it will not move faster, so you have a super shift. But the goal of the experiment is to see if your protein is binding to your DNA. If you want to see if your protein is binding to your DNA, you should be tracking your protein. So the antibody should be raised against your protein and not the DNA. Was it clear?
Yes, yes, I got it, sir.
Okay, right. Okay. Next question. It could be a continuation. During gel mobility shift assay, DNA protein complex is resolved on option A denaturing polyacrylamide gel, option B horizontal page, option C non denaturing polyacrylamide gel, option D 0.8% agarose gel. You can think about this for a couple of minutes and justify. Okay, again we go with eliminating the incorrect options. You can start by eliminating the options that you think as incorrect, and then we'll come to the right option later. Okay. Yeah, does anyone want to start with eliminating the wrong options?
Sir, we can eliminate first and fourth.
Okay, why would you want to eliminate first option?
First, because denaturing gel can disrupt the DNA and protein complexes or their structure.
Right,
And
good.
0.8% agarose gel is like large pore size.
Okay, if you have a large pore size, what would be the problem?
Then resolution will be not that good.
Right. Yeah, so these two are incorrect. Good. What about the other two options?
Horizontal also we can remove, but I don't know the proper reason.
Right.
And C would be correct answer.
Yeah, so people, I personally have no experience with horizontal page. The thing with gel mobility shift assay is, uh, you want to preserve your DNA protein complex, which is why you can't use a denaturing page, which is why you will use a native page. A non-denaturing page is similar, a name that is used for native page, where you don't have STS or any of the chemicals that would denature your protein. Right. So option C is correct here. Good. Yeah. Next question. Again, it's related to one of the previous questions. Which of the following techniques is used to find genomic binding sites of a known protein? Option A, gel mobility shift assay. Option B, native horizontal page. Option C, DNase footprinting. Option D, 2D electrophoresis. Yeah. Does anyone want to talk about any of these techniques? It could be not relevant to the question, but you can tell what you know about a particular method here.
Again, it would be footprinting, sir.
You're saying the right answer could be DNA footprinting. Okay, DNA footprinting or DNA is footprinting? And if so, what would be the difference?
Difference in, sir, what, sir?
No, no. Yeah, are you saying the right answer is DNA footprinting?
DNA footprinting.
Right. Do you want to comment on the other options?
Yes, sir. 2D electrophoresis is the protein separation method.
And
And
what happens
there is,
in 2D electrophoresis? In 1D electrophoresis, we separate proteins based on their molecular weight. What happens in
yes,
the
sir.
2D electrophoresis?
On the basis of isoelectric point along with molecular weight.
Okay, what is isoelectric point?
This is related to something pH.
Right.
Where the charge is neutral.
Right. So your protein is chargeless. Your protein has zero charge. Yeah, right. That is your isoelectric point. Good. And gel mobility shift assay we had seen earlier. Right. I'll just describe briefly what 2D electrophoresis is. So instead of separating your protein by just its molecular weight, when you have a lysate, a total cell lysate, you can separate it on more than one basis. So here they use a method called isoelectric focusing, where you have uh, a strip with different pH here, from lower pH to higher pH. And as you pass through, proteins that have a particular pI, that is the pH at which it has zero charge, it will stop migrating. And then these proteins can be resolved on the second dimension by running it again in an orthogonal direction. So now they are also resolved based on their molecular weight. This is called 2D gel electrophoresis. Coming back to the right answer, it's DNA footprinting, although the option mentions it as DNA footprinting. So here what we do is we allow protein to bind to DNA, and then now we the idea is, because the protein is bound to the DNA and it is fixed, that part of the DNA will not be uh, cleaved by any of your nucleases. So when what you see as scissor here is a nuclease, which is a DNA degrading enzyme. So you have restriction endonucleases that you might be familiar with, that cuts DNA at specific positions. So when you add these different restriction endonucleases, this particular region of DNA will be free of any cuts because it is protected by your protein. And later that can be run and sequenced to identify what region is bound to your protein. Right. That is the method that people use to find genomic binding sites for a known protein. And in continuation with that question, they ask about the enzyme that is used in footprinting assay, and that is DNase, like we discussed earlier. Now, there are other options: RNase, DNA ligase, and DNA polymerase. Can anyone comment on what the other options are? Whatever you know about the other options can also be mentioned.
RNA is for the RNA degradation, and
Okay.
ligase is for joining, and polymerase is for polymerization process.
What does
That's it.
ligase do?
Ligase for joining of DNA segments.
Okay, is it a covalent joining or non-covalent? What exactly happens there? What kind of bond is formed when this joining happens?
Between the DNTPs,
Yeah, DNTPs is used. That can be used with your DNA polymerase. But why would you need DNTPs with your ligase?
it is required on the that lagging strand.
You need not go into lagging strand. There, that is uh, different. You have two strands, leading strand and lagging strand. That is different. In DNA ligase, what when you use DNA ligase, what happens? Is there any particular bond that is being formed?
It's a phosphodiester bond.
Right. So you have a phosphate, 5' phosphate and 3' OH. And as long as the bases are compatible, this phosphodiester bond is formed between these two. And DNA ligase helps in that. And it's an ATP dependent enzyme. You need to add ATP for it, this to work. DNA polymerase, on the other hand, helps in polymerization. This is what we use for PCR, which is polymerase chain reactions, where we want to amplify particular segments of DNA. And this is where we use DNTPs as well. Right. The next question is, DNA from fossilized samples is extracted and amplified with random primers. How will you determine the size of the amplified DNA? And four options are given here. By sequencing the amplified DNA along with DNA ladder, by analyzing DNA in mass spectrometer along with protein marker, by analyzing DNA in pH along with protein marker, by resolving DNA in 0.8% agarose gel along with DNA ladder. Yeah, so the question is, how will you determine the size of the amplified DNA? Very simple question. A very simple method is enough to confirm this or do this validation. So you can think about this and for a couple of minutes and then unmute. Yeah, so the question is, how will you determine the size of the amplified DNA? So what is the easiest way to determine the size of an amplified DNA?
Simply by using agarose gel electrophoresis.
Yeah. So that's the simplest way. So here, by size we mean the number of base pairs that is there. and when we have a DNA ladder with us, it will both will get resolved, and you'll be able to see what is the length of the amplicon. Right. The next question is, DNA is dissolved in non-denaturing gel while mRNA is dissolved under denaturing conditions because option A, mRNA is small in size, option B, mRNA has secondary structures, option C, mRNA is sensitive to RNase degradation, option D, mRNA has uracil in their sequence. You can take a couple of minutes to think about this question and then start with incorrect options. You can start eliminating incorrect options. Take a couple of minutes and then uh, we can start discussing this. Okay, so the thing with RNA, specifically mRNA, is it can form something called as Hongsteen base pairing. And unlike what we studied, that A and T forms two hydrogen bonds, G and C forms three hydrogen bonds, in mRNA it is different. It can form multiple different structures where G can interact with U, not just A with U. And when you have such a complicated set of interactions, your RNA can take multiple different conformations. It can form multiple bonds that you might not be directly able to perceive just from the sequence. And mRNA is single stranded. So when this is the case, uh, you will have more than one species in the same tube. What I mean by species is, In some cases, this G can bind to this U. In some cases, it might not. Or when this G binds to this U, this G can also bind to this U. So structure would vary. And if the structure is varying, when you run it on gel, you will not see one crisp band. Instead, what you will see is multiple bands that are fused and smeared. So this is why you need to linearize this. And you can't linearize this with SDS. So instead, what people do is they add something called as formamide. What formamide does is it interacts with your nucleobases here and thereby prevents any of these hydrogen bonds from forming. So it basically denatures your RNA, which is why you need to run mRNA in denaturing conditions to resolve it. So the right option here is mRNA has secondary structures. Okay, this is a continuation of what I said earlier. The question is on the denaturing agent used in agarose gel for resolving mRNA. So people use formaldehyde as well, but formamide is a preferred denaturing agent that is present in RNA loading dyes. We can't use SDS because SDS binds to your protein. Beta mercaptoethanol is something similar to DTT, what we discussed earlier. It is also a reducing agent. It will also ensure your disulfide bonds formed by cysteines are not present. It will prevent its formation. Whereas temide is what you use for polymerizing your gels. Right. Your next question is RNase free water is prepared by treating water with option A STS, option B DEPC, option C beta mercaptoethanol, option D formamide. So you can take a couple of minutes to think about this question, and then you can start mentioning what you think is the right answer.
Sir, correct option is option B, DEPC.
Can you expand what DEPC is?
Diethyl propyl carbonate.
It's actually diethyl pyrocarbonate. Do you know how it works?
Sir, it is added to water and then it is incubated for few hours, after which it is autoclaved.
Hmm. Okay, so I'll first mention why we need to prepare RNase free water and when we need to prepare RNase free water. Whenever you're trying to purify RNA from cells, you need to do this. This could be for processes like um, RNA sequencing, whole cell RNA sequencing, or for structural studies where you want to purify an RNA and then crystallize it, you will need this water. How you do this is you take your normal molecule water or deionized water, add 1 is to 1000 or 10,000 ratio of this DEPC. What this does is it binds to your proteins in the water. You will have RNases, that is nucleases and proteases, naturally in every place, whatever water you store. So, in order to remove that, you need to inactivate these nucleases. This DEPC goes and binds to lysine or arginine, those charged residues that are present in your proteins, and it inactivates them. That is why we use DEPC for RNA related work. The next question is the reagent used to enhance staining of an agarose solution is dash, specifically for RNA. Okay, so for enhancing the staining of agarose gel with RNA, people use ammonium acetate solution, and this is with when you use ETBR, because unlike your double-stranded DNA, here you would have denatured the RNA, so your ETBR might not bind really well. It might not intercalate really well. So to enhance it, we use ammonium acetate. The next question is the most appropriate column used to purify the labeled oligonucleotides for a gel mobility shift assay is dash. Option A Cefadex G50, option B Dionex Carbopac PA1, option C DEAE, Cefadex, option D Q Cefaros. Cepharos. Option D Q Cefaros. Option C DEAE, Cephadex, option C DEAE, Cephadex, option D Q Cefaros. Option C DEAE, Cephadex, option C DEAE, Cephadex, option C DEAE, Cephadex, option D Q Cefaros. Uh, you can think about this question for a couple of minutes, and then you can. It's easier to answer if you eliminate the incorrect options. So to start with, maybe you can eliminate. You can whoever wants to answer this can start by eliminating incorrect options, and yeah. Okay, so what we want to do here is purify oligonucleotides for gel mobility shift assay. So here the expected size is 30 to 50 bases. When you say oligonucleotide, it's a single strand. So you might have heard about ion exchange chromatography. Q-Cepharos is used for anion exchange. DEA Cepharos, Cephadex, either of these are used for cation exchange. Dionyx, it's written as carbo pack, meaning it binds to carbohydrates. So all these three options are incorrect. That leaves us with only Cephadex G50. Here, this determines the size. We have Cephadex 75, S200, and multiple different variations that determines the size of the pores, and hence the ability of that column to purify. So we can eliminate options B, C, and D solely because of the nature of these. Because you cannot use anion exchange or cation exchange to purify these oligonucleotides here, and definitely not a column that is used for purifying carbohydrates. Yeah. The last question for today's class is gel mobility supershift assay can be done by option A performing electrophoresis in high voltage, option B increasing the runtime of gel, option C introducing specific antibodies to the binding buffer, and option D you incubating with 5 folds more protein. So think about this question, and then we have discussed this earlier. You can take a couple of minutes to think about this, and then we. Sorry? Option?
Is it C?
Yeah, it is option C
Specific antibody.
with specific antibodies. Yeah, that's the right answer. Yeah. So with this, we come to an end of today's problem solving session. If you have any doubts, you can discuss that with me. I'll be on the call. Thank you.
protein and DNA
Huh.
is more stronger than bonding between protein and antibody? Like which
So,
one is stronger?
antibody is specifically raised against your protein. So that will be having the best binding affinity.
Okay, thank you, sir.
Yeah.